Okay, so here we go. We're back. So Luna, just hold on a few words before, even if you're really committed and ready to go. So welcome back, session 2. Just checking that the microphone is on this time. Seems to be. So this afternoon, guys, thank you for being here also, whatever you're still stressed or already relieved. This afternoon, we're going to talk about stuff like the world of clouds behind Luna here. So we've got blooms, MPA, Mediterranean Sea Management. The title of the session is Research and Management for the Mediterranean Sea. So we're going to hear about that thanks to Luna Lombart, Alexandra, Rubin, Joran De Gang, and Adrien Dutrey. Part of your jury. I think you know them. Aziz Moula is postdoc at Ecosys, part of Université Côte d'Azur. Adrien as well, research engineer from IRCAN. And with us, we also have Timothy Cook, science officer at Blue Seeds. So you cannot see Timothy from the public, but you will be able to see him from the point of view of the presenter. I would also like to thank the fantastic people who took the time to review your fantastic reports, for most of them. So I mean, Aziz Moula, of course, some of the other people that you know, for example, Steve Como, researcher at La Sorbonne Université, Luisa Manguelaggio, professor at Ecosys, Florence Lacrosse, no surprise now, science head now of Coral Guardian, Timothy Cook, Blue Seeds, but also Marie-Yasmine Botin, professor at Ecosys Université Côte d'Azur, and myself. So, with no further ado, Luna, are you ready?
So,
just check that your presentation is also ready. Yes.
Okay.
Enjoy for the next 15 minutes.
All right. So, hello, everybody. Today, I'm going to tell you the improbable story of a Mediterranean coral species called Cladocora caespitosa. So, when I talk about corals, you're probably thinking about this, right? And what about this one? Did you know that that one is found in the Med, right here in our backyard? And what about this colony? This beautiful, thriving colony was found in a port de la Rague in Mandelieu. Now, let me tell you a bit more about this coral. This coral is an endemic bioconstructor, meaning it creates reef, just like I showed you. It's endangered because its population is decreasing, but it's not protected. It's a keystone species, meaning it provides complex habitats for other species to thrive. And unlike other corals, it has quite a slow growth rate, and it's got a very low dispersal rate. It's got two morphologies. The first one that I showed you is called a bed. It's a ball. And there's the bank morphology, which is more encrusted. And it's a fusion of several colonies in one place. It feeds on autotrophy during the summer, when there's lots of light, and on heterotrophy during the winter, when there's low light. It's found usually between 5 and 40 meters on hard and soft substrates in both turbid and clear waters. And it prefers usually places with low waves, not a lot of waves, but still quite some hydrodynamic flow. So it's widely spread across the Med, and there were 31 populations that were observed. But on these 31 populations, only 2 were really studied, and that's in the Columbrit's Island, which is in Spain, over here, and in the Adriatic Sea next to Croatia, where there's a bank that measures over 650 meters square. So obviously there are major threats to these species, which we have talked about before, such as sedimentation, pollution, and marine heat waves. So it's a bit weird to find all these colonies in a port, right? So my question, I wanted to sort of think, why are they here, obviously? But is this really, truly an exceptional case of adaptation? And I want to look at what are the steps needed to develop a conservation program for these species. I'm going to focus mostly on the first question, and I'm happy to discuss the second question a bit more after. So, to answer this question, I want to look at several questions, sub-questions, such as what are the best methods to conduct this preliminary study and to ensure a long-term monitoring program with natural diet? Why is the Port de la Rague suitable for these colonies? And also, is Cladocora caespitosa capable of adapting to these challenging conditions? And how can public outreach boost conservation efforts? So, the study site, we have the Port de la Rague, which is in Mandelieu, And the main thing to remember with that one is that little blue line, it's a river coming into the port, okay? And also, one more thing to remember is that this little thing here is the petrol station. That will be useful for later. I used the port of Gare as a control port, which is approximately the same size, but there are no colonies because Nature Dive, the people that I worked with, went there and didn't see any colonies in the port. Also, there are no rivers coming into the port. So to answer the question of why the Port de la Rague is suitable for these colonies, I wanted to do some water sampling. At both ports, I did several sampling sites to try and cover the whole port. And what I did is I measured pH, temperature, salinity between March and November, as much as I could, during the same day or the following day, at the surface and at the bottom. So to look at, is Cladocora capable to adapt these challenging conditions? I wanted to look at different methodologies. What would be the best methodology to see first if they are there and where are they in the port? So methodology 1 and 2 are quite similar. I went in and measured each colony and I recorded biological and environmental parameters such as necrosis, bleaching, substrate, overhead, all those type of things. I located their position using an underwater GPS and took some pictures, some photos, just like these ones, for future analysis. And the second methodology, well, the first methodology involved both divers to do the same thing. And then the second methodology was we split the tasks and also we reduced the number of parameters recorded. The third methodology, we pre-mapped the area on a slate and we noted all the boat location, all the boat spaces, all the pontoon pillars, all the mooring blocks. And what we did is we went in the water, used an underwater scooter and counted all the colonies that we found along a 4-meter deep transect, imaginary transect. So this required the diver obviously to count the boat spaces, but the site was shallow enough to go back up to the surface in case they were lost. This allowed us to see the area with the highest density of corals, and therefore we wanted to do photogrammetry. And for that, we used 3 ground control points, just like you can see in this image. And with we created a 5-meter square quadrat and took pictures of the whole area. Now we wanted to do a coral nursery table to start some sort of conservation initiative, and we decided to locate it at the end of the pontoon there because it was following all these parameters, but also to relocate some colonies that were in dire need of saving. So now to our results, the water sampling. So remember, I'm trying to look at why is this port suitable for the colonies. And first, I looked at the salinity difference between the 2 ports. And with that one, we can see that there are no differences in between the 2 ports, but we can see that there's quite a lot of variability in La Rague. So it goes from 4 ppt to 43 when the normal is about 35 ppt in salinity in the sea. For the temperature, again, we can see a lot of fluctuation in the Port de la Rague. The difference was significant, and we can see that the temperatures range from 13 degrees to 27 degrees in the Port de la Rague, which is quite a big variation. And for the acidity, the overall thing that we need to remember is that La Rague was less acidic, but still more fluctuations. Now, my mapping, which was quite the big work of this internship, was so the first 2 methodologies were very, very time consuming and were not very efficient. In one dive, I measured probably 8 colonies, which was not good. But with the last method, in one dive of 3 hours, maybe, it gave us the global distribution of pretty much a lot of colonies. And that allowed me to create this map of approximately 200 colonies. And these areas right here, and here, and here, were not observed yet. And then with that, I could create a first density map using the port structures. And with that one, we can see clearly that the highest density is located right under the petrol station, which is right here, with over 35 colonies. And then with that, I created a beautiful map which I think using the inverse distance weighted interpolation to estimate the coral density within the whole port. Now, I thought this map was really beautiful. I'll be happy to talk about it after. And the photogrammetry, this is a photogrammetry of right underneath the control station. And we can see those yellow balls here are the colonies. So there are quite a few, they are quite small. It's not exactly the picture that I showed you at the beginning, but there are quite a lot of colonies there. And it gives us an idea of the substrate, the size, and measure their, not the health of bleaching, the necrosis, and all of this. Now, for the coral nursery, the corals that were saved, it was saved during that first dive, that natural dive did, but they needed to be relocated. So we took this opportunity to test the propagation transportation methods and to do some photogrammetry to see if the attachment method that we use is successful, if the placement of the colonies is successful. So now, the discussion. What makes a port suitable? So we know that ports have quite a lot of pressures, right? They have lots of water pollution. They degrade their habitats. They have some contaminants in the sediments. And this is quite harmful to the biodiversity, as well as extreme variations. They usually are characterized by this: extreme pH variation, temperature variations, and salinity variations. So the polar here, what we saw, is those fluctuations are present. They are there. There are quite a lot of fluctuations there. And also we know, well, I know, and I'm telling you now, that there are some high sedimentation rates because of the river. And for that, they need to do regular dredging. And the dredging involves the sediment analysis, ensure that there are quite some elevated heavy metals levels in the sediments. And corals are quite sensitive to all this, which, answering that question, no, it doesn't seem like a fault, is suitable for these corals. But there is a theory about the nutrient input from a hydrodynamic flow that may support coral survival. Now, is Cladocora caespitosa capable of adapting to these challenging conditions? There are studies that show that above 50% of the mortality, there was more than 50% of mortality in a population in the Ligurian Sea because they were exposed to temperatures above 24 degrees. Also, in the Gulf of Gabes, there was the implementation of a phosphate industry, which polluted the water, which caused a decline of the whole population there. But in some places, in the columbrates, they were exposed to temperatures above 24 degrees for more than 60 days, and they showed no sign of necrosis. Also, there was a study in Monaco by Christine, and she looked at the heterotrophy, and they increased the resilience in thermal stress in these animals in Cladocora. So my point here is that there seem to be different adaptations depending on different populations. And remember the fact that there were low dispersal rates. And this is because of their negatively buoyant egg. So they don't go far, meaning that the populations across the different seas are quite genetically different. And this is also seen by a population in Formentera, which are free-living nodules. They don't need any substrate to attach because they have competition with microalgae. About the conservation, the photogrammetry will help monitor, obviously, those conservation initiatives. And also the nursery table is the first step to understanding how we can create a restoration program. There are not a lot of restoration program on this species. There are actually none, one that is starting. So this is a good starting point, I guess. Now, I tried to do some public outreach through public surveys, through this one, which you guys are welcome to have a look at after. Not now, because you need to listen to me. It didn't go very well, and I'm happy to discuss it afterwards. Okay, and through local school workshops. And the aim is to increase the awareness and therefore create a conservation program. Now, there were some limitations to this study. For those who know, they will know. But more seriously, there was a fire in the building of the office. Therefore, we had a big gap in trying to collect the data, and that caused me to have irregular data. And also, I think there was a lack of tools. I think we needed some tools to have some regular monitoring of the data, such as the hobos. So there are little tools that you leave in the water and record all the parameters that you need on a regular basis. Also tools to accurately measure the nutrients and contaminants. And I tried that with a colorimetry test, aquarium test, and they were not sensitive enough for me to detect all these changes. For the grammetry, the resolution was quite low. I think we need more pictures and better pictures, such as macro and wide angle, probably mix the both, try some different methodologies, and a better computer, because my computer cannot deal with meta shape. Also, it was a very short time for the study. I think to look at those trends, we need to have a long-term study. I think we need to look at those trends and those changes, those fluctuations over a year, or even 2 years, to look, because there are different conditions that we need to take into consideration, such as extreme weather events and that kind of thing. And also 2 ports. This was limited to 2 ports, but I think it would be very interesting to find another port with the same characteristics, or a site with the same characteristics, and this compare, are there colonies there? Are they thriving? Are they the same size, etc., etc. Now, conclusion is an exceptional case. I think it is, personally, but only 0.01% of the ports in the world participate in research in biodiversity. So we don't really know what's happening in the other ports. So we don't really know if it's that common. But those extreme fluctuations that I have noticed during this study showed that it might be still an exceptional case, because over 200 colonies were found in this port. And my main theory hypothesis on this is that the river inflow is what apports nutrients and the hydrodynamic flow needed for the species to thrive. I should thank everybody, the Maris team, Natural Dive, and the Port de la Vague management team, and thank you for listening.
So in a minute, Aziz is going to ask first question from the jury, but as promised, the first question comes from
why do you think this type of corals are worth saving?
What about you? It's important to do this.
Well, I think that's what we don't realize, is that we have, we're all talking about all these coral reefs in the tropics, just like this morning, and they have this big role of being carbon sinks and to bring tourism and to bring those ecosystem services. And probably their job here is not as big as the job they have in the tropics, but it is, what we don't realize is that we have that here, right in our backyard. And it could be the same level as what they provide in the tropics if we maintain them, if we protect them, if we conserve them. And I think people are not aware enough of their presence, of the fact they're out there. And it could increase so much stuff. It could increase the tourism, it could increase other ecosystem services, like the carbon sinks, like coastal protection and all those things. So I think it could be very, very valuable if we put more value to it.
Thank you. So here we go to Aziz and Adrien, and we'll finish with Timothée for this one.
Super cool. I really like this topic. I have a question, and I'm going to sound like a hypocrite now because I told you to delete it from your thing. But you mentioned it about the reproduction as well. That's a question on it. Do you think I think you said the reason why you think there's 200 in here is because the environment is suited for it. Do you think there's any other reason to do with reproduction, as in when this reproduces, it has a clustering effect because the egg is negatively buoyant, it stays around in the colony? Do you not think it's kind of like a chance-like event where there's just clustering, because we do see this with larvae all the time. They cluster together, and as they grow, the density becomes really important. Do you think?
Yeah, absolutely. I think that would be one of the reasons that they are more. Also, I think because they also reproduce by fragmentation, and I think the chains and the anthropogenic activity in the ports has been like breaking a lot of corals, and they do also those cleanings every year in a port where a lot of divers go in the port and just clean everything, and they don't realize. I mean, I've done it myself, and I've seen the divers, and they just break. They just break everything, which is bad to say. And I think, I think the fact that they have a low dispersal and the fact that they can break and just go on again, then will be one of the reasons that there are so many. Yeah.
And you mentioned you measured this by density.
Yeah.
Usually for corals, and I'm happy you did it by density, but usually for corals we do coverage. But obviously for this particular model species, it's a pretty difficult thing to do coverage on a small area. But how do you think density helps to understand the resilience of this species?
How do I think density will help the resilience of this species?
Not help, but understand. Yeah, instead of taking cover, for example.
I guess it's hard to do the coverage because, I mean, just the fact that they are more balls, more just like separate colonies rather than a big cluster. Like I showed in the photogrammetry, we can see that it's like balls. But I mean, the fact that we saw, like we counted how many colonies were in one place helped to like determine why this place could be more beneficial than the other. I guess I'm trying to answer your question, but
no, because I really think I'm kind of advocate of taking density rather than coverage is really important. So I just wanted to know why you decided. Was it just because there's not that many there, and there's a lot of empty space? Or
well, yeah, I think that was the main reason, because it's not like the pictures that I showed you at the beginning. It is really just like colonies and colonies and colonies, but there's still quite some space around.
Okay.
There were some places where it was started to be a bit bigger and therefore form like a bank, like I said, like a fusion of different colonies, but it wasn't enough to say again like the coverage. But I think it would be interesting to focus also on these big colonies and study them a bit more and understand.
Could you go back to the map?
Yeah, which one?
Distribution.
If I may, just one thing regarding the coverage. So I think you did great, because the thing is that addressing coverage in this type of environment, it's completely biased because it's not an open space.
Yeah.
So I think that you cannot compare coverage of a pore than
100%.
So I think I think it's maybe also the reason why you pick also the density.
Mm-hmm.
The widespread, where the studies. I'm sorry.
This one?
No, in your introduction. One with the whole Mediterranean.
Sorry, that's my bad.
So this is based on studies. Yeah, studies. Okay, so it's pretty widespread across the Mediterranean. It's endemic to the Mediterranean. It's the only coral. But I want to know: is there any previous data? What are these? Just observations, or are they studies, or what is the data from them, and how historical are they? Do we see a change in the abundance?
Yeah, actually, we do. This is a literature review of everything on the Cladocora that was found. So these are all human observation and studies. But I've seen using the GBEF data thing that actually Cladocora was found elsewhere in Africa as well before. It never stayed there for some reason. I didn't really look into it.
In the Red Sea?
In the Red Sea, I'm not sure.
Or where?
In the south coast of
okay.
of Africa. But I think there was it was not a lot. It was not widely spread. It was like a few sightings. So the reasons why they were there, why they're not there anymore, I've not really looked into that, but they were seen elsewhere before, but way before. Yeah.
Thank you very much.
I didn't then.
So I will not evaluate you, okay? Because I'm doing so much with you regarding this project, but I have the right to ask you a few questions. So do you think, because do you think that if a coral is growing really slowly, it can invest much more energy in defense and then in resistance and resilience?
Well, I feel if it's growing,
Because you say that it's a slow growing.
yeah, very slow growing compared to Aquaphora or something. It's only like 1 centimeter, a few millimeters to, anyways, it should have printed them. But I feel like if it's growing that slowly, it's because it needs quite a lot of energy to grow and therefore wouldn't be able to put that much energy onto the resilience. But that could be one way to see it. Or maybe they just don't put that much energy into the growth and therefore can put more energy into the resilience. I mean, it could be seen both ways, I guess. Now, which one I would interpret and which one I would should, I'm not pretty sure. Which I understand, they do rely a lot on that heterotrophy. So I guess they have like enough energy to do that and to use that energy to stay alive.
It's in a very stressful environment. So yeah, sedimentation,
Yeah, no, but that's my next question is that maybe a next step also will be to look at, I know that the lipid composition of coral can give them much more advantage in resistance in temperature or other things. So maybe it's something you can also compare now with this study and start to investigate. So because I think that this is something that can give you insight about why this one specifically raises so much in this.
I mean, there's a lot that we can look into more into, like inside a core, like you say, the lipid concentrations, maybe the genetics, that could be further studied to sort of look at why this population is there.
And one more question. So you mentioned in your data set that the pH in this specific zone, while the salinity is stable, then the pH vary a lot.
Yeah.
And what we
that is correlated.
hypothesis that is it correlated with the fact that these animals are more resistant because they are more exposed to this pH variation? So, and then they become more resistant because they are challenged. I mean, much more than some other.
Yeah.
I don't know if you have any literature example or if it was studied within this species.
It was studied within this species with a different acidity variation. From what I've seen in the literature is that as soon as like there's too much acidity variation, then it causes obviously the bleaching. And I don't know, I didn't mention it in the talk, but it actually went down really low in La Hague. It went down to even 6 pH, which is I mean, very acidic. So I assume that the fact that that's why I was saying also that long-term study is like much more needed to see if those fluctuations happen all the time. And therefore I would think that because they are exposed to those variations often, always, then they must be more resilient.
But do you know if in Volcano Island, in Eolen Island, is there this species or not?
No. They're not too sure, but I haven't read about them in the Volcano Islands.
Okay.
So I think I would have because like there's not that many studies on the Cladocora. So, but yeah, I didn't specifically look about that.
Thank you.
Thank you, Adrien. Timothée, your time.
Hello, thank you very much. I really enjoyed this this presentation, and I don't really have any specific questions or even advice. Maybe just take these as comments. But I think that considering the complexity of the question, you did a really good job to explore the different reasons why there would be such differences and the the parameters which might be able to predict why it's more successful in one area. And you've set a certain number of hypotheses. I've understood there's basically, let's say, genetic hypothesis, and the other one would be this river hypothesis, but you don't exclude other factors. I was quite, I really also like this because I actually did my PhD on similar questions, but with a different model. Seabirds subjected to different conditions. And so, after a PhD, you think, you'd have enough time because you were talking about time as a solution to unravel these different questions. And I think that's what I love about science, is that even after 3, 4, 5 years, you still don't know. But there are certain ways to address, in your case, which might be actually easier than for a seabird, which is long growing and just doesn't grow or breed as quickly. You mentioned longer time series of measuring those variables. So yes, and actually trying to compare how temperature, acidity, and so on varies over time, and having on your nurseries follow the growth patterns of those corals in relationship to the variation in the other variables would probably be something that would help. Yes, this whole thing about different populations and adaptation, natural selection to local conditions, I think it's actually underestimated how species are able to genetically adapt to local conditions. At the same time, we also fail to recognize how plastic they are and how they are also capable at the same time to respond to a great variability of conditions. And there's something, it's called cross-fostering. So you take individuals from one population and you make them grow in the other population. And so in this way, you try to untangle the effects of the environment and the genetic background. But again, that's, if you get a PhD, then maybe you can do this. So I don't know. As I said, these are more comments, but good job. And I felt you had given quite a good thought to all these different questions. So I have no question here. Thank you, and well done for the very interesting work.
Thank you.
That's quite quite rare. Timothée has no questions. So in this case, I will take my responsibility. Especially so here you were working with Nature Dive, so it's an NGO. I have maybe a general question. It could correspond to any people who are working in NGOs. So here you're really working on scientific data. So how do you see that in the future? So for example, who could be the partnership that could be developed from Nature Dive with another entity to ensure the quality of the science behind that? Because in Nature Dive, if I understand correctly, there are a lot constituted by divers who are not all scientific. So what's your point of view on that, and what do you think?
Well, I mean, the main people in Nature Dive, so Sam and Damien, are scientists themselves. But I think what would be good for them, because they are very ambitious and have a lot of projects, and I think it would be good for them to have to keep some sort of partnership with some students that are interested in carrying on this project. I think it would be interesting to have maybe a PhD student to do that over the long term. But like I mentioned, there was this woman, Christine Ferrier-Pages in Monaco, which has been studying the corals, Cladocora. And I have contacted her, I have talked to her, and they were willing to help Nature Dive do this more chemical testing and testing of the chlorophyll in the water, those kind of things, because they had the tools to do so. So I think partnering with some big facilities like this, like the Monaco, or maybe even in Ircon, to that's forward. But I think they need a person in the association in the NGO to focus only on that but because I think it deserves a lot of time. I think it's a beautiful project, but I think it deserves a lot of attention. And
Also, you were talking about this citizen science project in 30 seconds. Yeah, because frustration is not good, really. And you say, we can come back to that if we want. So let's come back to that in 30 seconds if you want. What went wrong, or what was most difficult? In
with the things I gave you, so I tried, my idea was to sort of set a public outreach program. I wanted to know first how people would react to this, to learning about all the corals being underneath their feet. So when I'm talking about the public, I'm talking about the port users mostly. So I made this little thing to try and get their attention and to also make them answer a public survey that I made. But I might have not been approaching them the best way because maybe I've said some things like could be accusatory. I did not. I said, did you know there were corals under your feet, and would you like to know how you can help them? But I sort of maybe implied the fact that they weren't like doing the right things already, in that sort of manner.
Defensive.
Yeah.
On that.
Probably, maybe. So that's the idea. I think I should have been a bit more open. But some people were interested, but somehow it didn't go through. And I think what would be very interesting is somehow get that word out, get those people to participate and get this awareness about the coral much, much bigger, and bring maybe local school, because Natural Life is working a lot with local schools, and to bring projects on the coral to these schools, such as like showing them how to propagate them, how to attach them, how to do those restoration techniques.
We're on both pages. Good. Thank you. I'm sorry we have to jump on the next one. I'm sure you will have time to explain that. So now you can. Are you ready for the next presentation? No. I do nothing.
Yes.
Yay!
Good luck.
Thank you. Just the water really quick.
Question is ready for Ali? Yeah? Okay.
We just use this.
Yes.
Go through it.
Just by the way, the keyboard as usual, and that's this camera today.
Okay. Good. Alrighty. Okay. So, hi everybody. Good afternoon. I'll be talking about assessing the anthropogenic activity on Posidonia oceanica here in the Mediterranean Sea. So, firstly, here we have a wonderful photo of somewhere that we all hold near and dear. We've all been here together, or most of us have been here together. We got here by boat, and looking at Laurence and looking at a photo, it gives us a good memory. But what we might not realize, and if you see in this photo, and we zoom in, a lot of these boats here are over sand in these light areas, but are also over seagrass. And so when we go to Laurence and when we're there, what we don't realize is what's going on underneath the water. So a lot of times these boats are anchoring over seagrass, and this is really the reality that we don't see. When the anchor comes up, it pulls out the seagrass from the sediment, causing a lot of damage. And so why does this matter? So to back up a little bit, seagrass is one of the 3 blue carbon ecosystems. They include mangroves, tidal marshes, and seagrass. And the ocean is able to absorb 20 to 35% of our greenhouse gas emissions, so that's wonderful. And a lot of that is due to these 3 ecosystems themselves. These 3 ecosystems are able to sequester 30 to 50 times more. They're more efficient than terrestrial ecosystems. So I know we're always hearing about the Amazon forest and how important trees are, but these 3 ecosystems are really vitally important as well. So I know we all know about Posidonia oceanica, but to give a bit more context, so Posidonia is found here in the Mediterranean Sea. It's our endemic blue carbon ecosystem. It is found in soft bottom sediments in tropical areas and in temperate areas. And it can be found up to 40 meters in depth. So here we can see in this photo, one of the amazing ecosystem services that Posidonia provides. It's a really wonderful nursery habitat. And also, as you can see here, Posidonia really protects our coastlines from erosion and from reduced wave energy. It can help in nutrient cycling, and it also sequesters 70 times more than a tropical forest, which I thought was really interesting. It is great for water oxygenation, and it can store carbon for millennia. So back to Laurence. When we are all snorkeling or scuba diving or just in the water and playing, this is typically what we see, but what we don't realize and what's so vital is what's going on underneath. So here we have really a schematic of Posidonia, but the whole Posidonia. So we have the leaves on top, and then we have the rhizomes, and then we also have underneath this part here called the mat. And the mat is really what's responsible for being able to take the carbon and sequestering it for thousands of years. So unlike terrestrial trees, the trees that we have outside, they take carbon and they put them into their bodies, for lack of a better word. But when they die or they get cut, that carbon is re-emitted into the atmosphere. Where with Posidonia, they take the carbon, they put it into the leaves, and they store it into the mat. And so even if the Posidonia may die, that carbon is sequestered there for millennia. But like I mentioned before, this is becoming a problem with a lot of anchoring that's going on. And so the anchoring not only hurts the leaves of the Posidonia, but it also takes out the rhizomes, and it can start affecting that mat underneath. So with this destruction of the mat, some of that carbon that's been there for so many years gets actually re-emitted into the ocean and into the atmosphere. And this is really a huge problem here in the Mediterranean. As we see, most of us have been on a boat since we've arrived here, or at least we've seen a boat since we've arrived here. They're everywhere. And there's a huge knowledge gap for the anthropogenic effect of what we are doing to the seagrass. So in order to assess the carbon accumulation rate, we need to look at in order to assess the sequestration potential, we need to look at carbon accumulation rate. And so carbon accumulation rate, we look at sediment accumulation and dating. And so carbon accumulation rate is the amount of organic carbon accumulated per unit area per year. So we can see how the sequestration is changing over time. And so by looking at time, we usually use 1 to 10 dating, and this is used for sediments deposited within the past 100 years, give or take. So this brings me to my research question. Do anthropogenic pressures, in this case boat anchoring, have a negative effect on Posidonia's ability to store carbon? And I have hypothesized that in areas where there is more boat anchoring, there will be less carbon stored there versus in areas where it's protected. So I did my project with the Laboratory of Oceanography that's in Villefranche. They're kind of like a pilot site of the Sorbonne, and we worked closely with the IAEA in Monaco. Here is our project site. It's in Cap Martin in the Natura 2000 zone. And so we wanted to look at a gradient of anthropogenic pressure. So here on the left, cores 1, 3, and 4C in green are areas where small boats are currently allowed to anchor. But then we have the site in the middle in purple. And this is where boats used to be allowed to anchor, but for the past 5 years, they have not been allowed. So we're calling this a recovery site. And then we have in blue here, the 3 cores, 5, 6, and 7B are fully protected. So we have anchoring, recovering, and protected. So field work. So here we have Steve and Samir about to go on their dive. And so they're taking these PVC cores that you can see, and they go underwater. Each core or each of the 3 sites was about 17 to 19 meters in depth. And they gently hammered the cores into the sediment and then brought them out. We taped them and capped them and then brought them to the surface. Brought them to the lab, and I cut open every core. So this is kind of like very classic what you would see, just a bunch of wet mud inside of the plastic PVC. And so I sliced them and made individual segments. So for the first 20 centimeters of the core, it was 1 centimeter slices. And after the latter 20 centimeters of the core, it was 2 centimeter slices. And each slice was weighed for its wet weight, dried at 60 degrees in the oven, and then until it was a constant weight, and then it was ready to be analyzed. So then we looked at the carbon, hydrogen, and nitrogen analysis. This part is still ongoing, so I'll touch on that in a bit. And then our friends at the IAEA in Monaco and another friend in Australia helped us to look at the lead-210 dating, and they used alpha spectrometry, gamma spectrometry, and modeling. So, like I mentioned, the carbon analysis is still ongoing. This project is still happening right now. Steve's current PhD student is taking this analysis as we speak. So what I did was, instead of using that data, because we didn't have it quite yet, what I did was look into literature of different sites here in the Mediterranean of Posidonia from 75 different sediment cores to come up with a % organic carbon value that I could use here so I can give you guys some results. So we have a lower bound of 1.8% and an upper bound of 6%. And from the few cores that we have looked at so far, our average value is about 1.5%, just to give you an idea. So still somewhere in that range. So results. Here we have the lead-210 in depth results. So this is a fitted regression line of the lead-210. So here are all the individual points, but also grouped by averages. The blue is the boat anchoring, the green is fully protected, and the red is recovering. And so here is as the core gets deeper and the amount of lead that's in it. And so we could see from running statistical tests that there is a highly significant decrease of lead with depth. And then we also saw with statistical analysis that the 3 different sites had a difference between one another. So to discuss this, this is what we expect to see. As you can see here in this schematic, this is from a past study, and this is where you see depth, and this is lead-210. So what we typically see is that, yes, with depth, there is less lead-210, and so this is following typical trends. And this is because deeper sediment is older sediment, and that lead is really decaying over time. So there is atmospheric lead that is being deposited into the ocean and settling to the sediment. And then there's also lead that's from sediment itself. And so when you're at the top of the core, you have both of these atmosphere and sediment sources of lead. And as you go deeper, that atmospheric lead decays. And so you're just left with the sediment lead. So you have less. We also saw that there was a difference between the 3 sites. And so our boat anchoring site had the least steep slope. This may be because of mixing at the surface, and the deeper layers is causing redistribution of the lead itself. For the protected area, we saw the steepest decline. And so this is showing undisturbed sediment, and it could be because this is a more preserved natural decay of lead. And then the recovering site was the intermediate. And maybe it's showing some residual impact from the past, but it's starting to recover a bit. So it makes sense that we would see it in the middle. But we definitely need to consider other factors like sediment mixing, erosion, and environmental conditions. And that would provide a more complete understanding of this. All right, so now from our lead understanding, we were able to calculate the sediment accumulation rate. And so here are the results for the sediment accumulation rate. So the recovering site is here, boat anchoring and fully protected. And from a first glance, you might think, okay, they look different, there's some differentiation. Well, the statistical test says that there is not. So there is the same amount of sediment accumulating at each 3 of the sites. So this is interesting and maybe contrary to what we would think, but this can be explained by, 1, the complexity of the sediment dynamics. There's a lot of anthropogenic factors coming into here and environmental factors like hydrodynamics and natural processes and sediment supply. So that could be part of the reason why we're not seeing a very clear trend. Another reason could be due to the compensatory mechanisms of the seagrass itself. So Seagrass is able to maintain its sediment stability very well. It is able to trap particles and has very good sediment retention. So it's possible, and I remind you that in the areas where there are boats anchoring, they're just small boats. So maybe their impact isn't that great. And also maybe the Posidonia's ability to trap particles and its good sediment retention is able to buffer some of those impacts. And that's why we're not seeing that much of a difference from the other sites. And additionally, this is quite a small sample size. This stuff is expensive and it takes a really long time. And so we only had 9 cores, but that has lower statistical power. So also some of the cores had very high standard deviation. So a larger sample size would be needed to have better replicability and better understanding. So finally, with the sediment accumulation rate and the % carbon that I found from literature, I was able to calculate the carbon accumulation rate. And there was found to be no statistically significant difference between any of the 3 sites. So where there's boat anchoring and where it's fully protected, they have the same carbon accumulating there. So that is interesting and misaligns completely with past findings. So here we have Pergent et al. They found a significant negative correlation between anchoring pressure and carbon storage, as we would all probably assume. So why are we not seeing this? So there's a huge limitation of the study. That is, I didn't use the % organic carbon that we actually measured. This was not used to calculate my carbon accumulation rate, and so it's from literature, and so therefore it's probably really skewing my data. How is it skewing my data? Well, there could be different % carbon or organic carbon from all 3 different sites. Each 3 different sites might have their own % organic carbon, and I use the exact same one for all 3 sites. Likewise, the % organic carbon may be different at the top of the core than at the bottom of the core, and I use the same for the entire thing. And so, yeah, the direct measurements of the % carbon, which is being done right now at the lab, is going to be analyzed, and hopefully with that accurate percentages, we'll actually be able to see a different result. So to conclude, so this study, we really looked at 3 different sites off of Cap Martin from different anthropogenic pressures to really understand and establish a historical carbon storage pattern. As I just mentioned, my initial hypothesis was not correct, which may be due to the main limitation of my study. And as it is an ongoing study, hopefully this hypothesis will be proven correct once Steve's new PhD student does all of her work and uses the actual data. And also future studies may want to incorporate nutrient input or hydrodynamic data to better explain the carbon and sediment accumulation rates and the lead-210. So going back to ailerons, when undisturbed, Posidonia oceanica is a very, very effective long-term carbon sink. It has a residual time or residence time of 4,000 to up to 6,000 years of carbon stock. But unfortunately, like we've all seen when we were at ailerons, and this photo is showing you, so much of the Posidonia is being affected by anchoring, so much that over a third of the meadows in the Mediterranean have been lost over the past 50 years. So hopefully studies like this, once it's complete, and other studies that are happening right now can really help bridge this gap and to better understand how our pressures or the anthropogenic pressures are affecting Posidonia and to provide a foundation for informed conservation. So thank you for listening.
Thank you, Ellie. So as we've started the other way around last time, let's start with you, Timothée, if you agree.
Do I have the choice? Thank you, Alex.
Timothée, hold on, I'm so bad. I'm sorry. Mila, you ready? You do have a question?
Yeah.
You ready, Ali? Because sometimes the question from Mila.
Right.
Since Posidonia is so important for carbon sequestration, and if you had to choose, do you think it's best to restore Posidonia or protect?
To allocate money for one or the other.
Sure. No, I think that this is kind of a big issue that's going on with like blue carbon right now is that we are like, oh, blue carbon, these ecosystems can just, take all of what we're doing and all of our greenhouse gases and fix it. And so there's carbon offsets and all this stuff. But I think really the issue is to stop the source. So I think these types of projects are important. So we like really understand why Posidonia is so important. But I think protecting it more so, not just like restoring, but really protecting it from the source, like what's causing it, which really is like the coral girls were saying earlier, like we need like policy change and, with less greenhouse gases and anthropogenic pressure and stuff. So I think, really protecting them from the source is the best way to go.
Protection first.
Protection first.
Okay. Let's go to you, Timothée.
Thank you. Thank you, Alex. That was really interesting, and good job on working on such a complex question. We actually had an intern from Mares Fernando Ruiz who a couple of years ago did a study on Posidonia and anchorage pressure. You might have read his master's thesis. No, well, that's the problem with master's theses which are not published. Hopefully it will be published one day, and perhaps not too long. Anyways, the take-home message was that one, as you mentioned, it's very difficult to get those samples, time-consuming, money-consuming, energy-consuming. So sampling is rather limited usually, and considering that there's a strong variability in carbon concentration between meadows and depths as well, it's quite difficult to conclude. So it can be quite frustrating, and I can understand how frustrating it may be to be working with limited sample sizes. Having said that, I was more wondering, because I often wonder about these questions myself, how do you see the link between anchorage pressure and meadow functioning? When I say functioning, I'm referring to their capacity to sequestrate carbon, because say if you have 10 core samples to throw into a meadow that's been anchored, wouldn't your results really depend on where those cores are dived into the meadow? I don't know if you see what I mean. So how do you explain that functional link between carbon sequestration capacity and the results you were looking for?
Okay, I'm not sure I understood everything you're saying, but I'm going to try to explain it. Stop me if I'm going on a tangent. But from looking at pathometry and what we did, we really tried to homogenize the methodology so that when we were at a certain meadow, we were trying to take the sample not from the edge, but more from like inside the meadow itself at each spot, so that there would be kind of like a continuous gradient or whatnot. And then you're asking how does anchoring really affect the sequestration capabilities of the Posidonia? Or I'm not sure if I caught the last part.
Yeah, that's pretty much it. Because if your meadow's been highly scarred, then visually you will see that effect, and so you're assuming that your core will also reflect that. Whereas if it's moderately scarred, I'm wondering what the difference is between a healthy meadow, depending on where your cores are driven in. So that's my question.
Sure. Yeah, I think that obviously it depends on how severe the anchoring scarring, like you're saying, is at a certain meadow. I know that when there is heavy pressure there, and there has been a decent amount of anchoring, and it rips out not only just the leaves, but the rhizomes, and it starts to get to the mat as well. It creates these like really big dead zones, and then other invasive species are able to settle there. And so it kind of breaks up the meadow itself and causes it to be kind of more patchy. And so I think that this could be probably seen in dating. Maybe you'd see like a stable level of carbon for the past, however many 100 years, and then it might start to get a bit discontinuous or mixed or fluctuating. So I think that you could probably see some of that in the dating. Does that answer your question?
Thanks. Yes, it does. I guess this takes me to my last question. So for that protected site, for how long was the meadow protected? And do you know how heavily, let's say, was scarred prior to protection? And, you know, considering how slowly Posidonia grows, what were you expecting from that site in terms of results?
Yeah, so originally we wanted to look at just satellite imagery in order to figure out where to take our samples from. And so that proved to be really difficult because of quality, resolution, and everything in the water. So we partnered with a local agency. They're called SMIAGE, SMIAGE, but in French, I don't know what that stands for very well. But they have been monitoring and going out to these sites in Cap Martin for the past 10 years. And they had a lot of data on that for the past 10 years. And so that's why we chose these sites. So from the past 10 years that they've been looking at it, I know that the site has been protected for at least that long. And the other site has been open to anchoring for at least that long. But yeah, I think that the site that was protected was like very healthy as opposed to the other sites that were lost.
Thank you.
No problem.
Thank you, Timothy. Sorry, I had to fix an emergency at the same time, but
No worries.
so let's go to you, Aldine.
Thank you, Ali, for this nice presentation. So, next question. So, why did you choose this specific site?
Yeah, so like I was just kind of mentioning, and I didn't put it in the methodology, but so for the first about like a couple of weeks of my internship, I was really looking a lot at satellite imagery because we wanted to look at where we could visually see a differentiation of the past coverage of Posidonia, somewhere that was used to be having a bigger amount of Posidonia, and in the past like few years, the Posidonia has gone a bit less, to be able to see if there's a difference there in the carbon dating. And from looking at the satellite images, you couldn't see hardly anything. Like it was really, really unclear. So we had to kind of pivot in February or so. And Steve had a contact at this entity called SMIAGE, SMIAGE. I'm not going to try to say what that is in French, the name, but and they had a woman who's working on a project at the Cap Martin site in Natura 2000, and they have been going out there themselves to take data and just to monitor those Posidonia. And so she knew confidently and had past like data logged. This is a protected site. This is a site that was 5 years had boats anchoring for the past 5 years hasn't, and this site allows boat anchoring. And we're like, well, that's perfect. And like we have, we trust you, and you have been looking at it firsthand. So then we chose those 3 sites just really to give the best gradient of anthropogenic pressure, just to look at kind of.
But in front of the marine station in Villefranche-sur-Mer, you have this crazy gradient. So why you did not choose to study these sites right in front of
I think that, yeah, it really was more so that, I guess, I don't know, Steve, we decided to look at Cap Martin just because this person had been there and she had seen it and they'd been logging it, and she knew that there was like differentiation there, and there was that gradient of anthropogenic pressure, and Steve didn't seem to know exactly more confidently about that in the Bay of Villefranche in that area. So we chose the other side because it was more knowledgeable.
And regarding this, the sediment collection.
Mm-hmm.
So to what depth do you need to dig to really and to collect to have data that are quite accurate for the site?
Yeah, so they went down, and they didn't dig anything, but the cores themselves, we made them like spiky at the bottom, so that, and they took a hammer, and I don't think, as I wrote gently hammered, but Steve laughed at the fact that I wrote the word gently. I wasn't there. I was on the surface, but I think like really went with the hammer, just for the depth of the core itself, which is about 100. I think the core is 100 centimeters, but when we pulled the cores out, none of them were 100 centimeters. Most of them were about 0 to 60 centimeters or so.
Okay.
was how much sediment that we obtained. There's a lot of trial and error. Some of them, the cores we took up, and it was just water.
And I have maybe a last question regarding one of your discussion slide. Okay, page, I think it's the 21. You say, okay, side comparison, boat entering equal less steep slope. And you explain why it's less steep slope. So, but for you, so all this information, what does that mean regarding the biology of the ecosystem?
For the lead 210, what you can see the lead. I think, I mean, it's not proven. It's just my kind of like understanding, and I have quite a limited understanding of lead 210. If I'm being very transparent with you. But what I understood was that you expect to see a pretty steep decline of the amount of lead at the surface to the depth. And so because the boat injury site was less steep, my assumption as to why we're seeing that is just because there's so much mixing of the sediment that maybe the mixing is causing there to be some lead coming back up, coming back to the bottom and such. And so that's what's kind of causing this more shallow decline of it, where you would more expect it to be, it's supposed to be exponentially. So it should be more quite steep. And you are seeing that steep decline in the protected area as opposed to the boat anchoring. But I think this is really just my total assumption. And you would have to do a whole experiment just on this or ask an expert on that 210.
Okay, thank you. I'm fine with the question.
Francis, you can pass.
Good talk. Thank you very much. I have a question about the sedimentation record, because you're a sediment core expert now. So I can ask you this. How would an anchoring like an event show up in the sediment record?
So it's complex. So even I was looking at literature, and they were saying like the complexity of sedimentation, like the dynamics of what's happening in sediment, is still in some of these literature. They're like, we still don't understand it fully. Like we still can't completely like tell you why we see more sediment accumulating here than not. There's hydrodynamics going on. There's erosion happening. There's maybe river input going on. There's other anthropogenic effects. There's so much like going into it that I don't think confidently you could.
It'd be pretty cool if we.
It would. It would.
What was the distribution of Posidonia in the sites that the 3 sites that you look at? What was the original distribution? Do you have any idea of the abundance of Posidonia?
When you say original, like
because you had 3 types of sites. You have one that's allowed boat anchoring, one that's fully protected, one that's recovering. Did they all have the same abundance of Posidonia before they became these treatment?
I'm not sure.
I'm not sure.
I'm not sure. Yeah.
Because one of them you said has been protected, recovering for 5 years. So maybe you have that data. Like,
yeah, the people that we partnered with, they definitely have all that data.
Yeah.
But I
okay. Yeah, it's a shame you don't have the direct carbon.
Yeah, it would have been much more interesting. But maybe whoever the intern is this year, because they're literally still doing it. I got the lead 210 data in October, November. I think November is when I got it, and yeah,
Well, let's say you did have them, and do you think they would follow what the literature says?
I mean, I'm hopeful that they would. The lead 210 is following what we expect to see, and the specialist who analyzed the lead 210 with us, he said that this is what we expect to see. These results look good for the sedimentation and the lead 210. So I would hope that the carbon would follow suit, because they're all kind of based off of one another. They're building on blocks, kind of. But yeah, like I said, I think that there's probably different percent organic carbon in each different spot, maybe from the core. So I really think this is like a huge part of understanding the carbon accumulation that I
think when you get them, it would be good to then see the difference between the ones that you took from the literature and the ones that you took.
It would be really
a lot of carbon values are taken from the literature, and
yeah, and they may
not be so
cool.
Just one last question. On a global scale, like on our is this a big problem compared to other sources of carbon? Is it a huge issue? Just the anchoring, not the release?
I think, yeah. I think that a lot of the literature that I looked at was saying, I was looking at mainly just the Mediterranean for doing sediment coring, but then also some other places to do sediment coring for seagrasses. And they were listing some of the major anthropogenic pressures, and anchoring came up every single time. Like, I think it is really quite extensive.
Thank you.
Good. So we have a few minutes for a couple of questions from you guys. Here we go. It's this time. Yeah, Adrien.
Yeah, I don't know if you had some information about the enforcement of the rules for no anchoring in the zones you were working on. No.
I don't know, but that would be interesting to know if the protected site really is
Yeah, exactly. Yeah.
actually protected or not. Yeah, I'm not quite sure. But yeah, that would be interesting.
One more question. Okay, and last question from the jury.
If it became a law, for example, I don't know, that we can't anchor anywhere, I don't know what the policy is in the Mediterranean, but for example, like in the Red Sea, it happens, but it's not meant to. There is a law against it. What would be your proposed solution?
I think it's not so much that you can't anchor, it's just where you're anchoring. So like there's plenty of places that are sandy bottom that I think would have less, I don't know, I'm not going to say they're not going to have any impact, but they would have less of an impact than if you're anchoring on seagrass or something like this. So I think maybe setting up like mooring or like eco mooring and such. And then also if you have to anchor, just being more strategic and smart about where you're anchoring rather than just anchoring anywhere. Yeah, just having more of a, guided place to anchor.
Okay, I think we're good. There's a question, Sophia.
Might have been that you explained and I didn't understand, but why did you look at lead? Like, what is signifying the steady slope or?
Yeah, we looked at lead because that's what the protocol just like calls for. If you read like all the literature on sediment coring all the time, they're really constantly looking at lead 210. I think that it is just like it's a methodology that's been done a lot, so they know how to do it, and it shows really wonderful like radioactive decay. And so we are partnering with someone at the IAEA, and that's like his specialty is doing lead 210 carbon dating. So as I arrived to the internship, they're like, we are doing this, and I was like, great, we're doing this. And so, but yeah, I think that it's a really good way to look at the amount of carbon accumulation from the lead 210 in the past 100 years or so. It's a pretty foolproof method that they've come up with, and it works well.
Thank you, guys. Thank you, everyone. And congratulations again. So, voilà, vous pouvez ça. It's time for a break. We're going to have something like good 15 minutes.
Okay.
And at exactly 3 o'clock, so it means you have to be back in the room before that. At 3 o'clock, Joran is going to give you a fantastic presentation on Ostreopsis, followed by Adrien. 15 minutes sharp. Well, you know what, Joran? Let's be crazy and start early.
Ambitious one.
Okay, so we're back. Yeah, I'm gonna try again. Okay, so this afternoon for the last part of the session, so we're gonna have Joran De Gang, followed by Adrien Dutrey, to still continue to talk about the Mediterranean Sea research and management. Are you ready, Joran?
Yes, I am.
Yes, you are. Allez, let's go.
Okay, welcome back from the break. I'm Joran. I'm going to be talking about the effect of environmental variables on the interannual variation of Ostreopsis ovata, which is pictured here. It's one of its aggregate forms, which I'll talk about later in the northwestern Mediterranean Sea. So there's something happening on our beaches. It's not. There's something happening, but not on the presentation. No, there's something happening on our beaches. So every year beaches are closing, people are getting sick. This year, 800 people in the Basque got a report sick from going to the beach, and it seems like Ostreopsis ovata is actually the culprit of this. Now, what is this weird Latin creature? It's a benthic dinoflagellate, and the thing is, it's blooming, it's growing a lot, and its abundances and blooms have been rising in temperate areas and the northwestern Mediterranean Sea since the past years. It's found in biofilm on macroalgae, but also seagrasses and other hard surfaces like just snail shells, but much less. Most of the stock of the population is actually in this biofilm, but it can get detached and then be suspended in the seawater and form these aggregates that I showed before, or even be carried through the wind with these water droplets. Now, there's a lot of consequences when these dinoflagellates bloom. First off, in the human health, as I mentioned before, people get really sick. This can come from a flu, fever, headache, respiratory issues. And the thing is that it's spread through aerosol droplets. So you can get sick not just by swimming, but also by just passing by on the beach. And we don't really know yet what any chronic exposure does to people. There's also some ecological problems with these blooms. When blooms have been reported, there have been sightings of mass mortalities of benthic invertebrates, mostly sea urchins. And there's also toxins found in bivalves and sea urchins that we are consuming. But we don't really know yet the effects on food safety compared to this. Then there's also some economic problems. With these blooms, beaches can close. And also there might be some association with illness to beach that might be coming later. So this could have a very negative effect on coastal tourism and recreation, which in the northwestern Mediterranean Sea is a very important part of its GDP. So this raises the importance of studying the ecology of this microalgae to help understand these drivers that drive the bloom, so we can improve our mitigation and monitoring efforts of this to reduce these problems that I just mentioned before. So this is where my research question comes in, is where I wanted to look if there was any intervarial variation in the bloom phenology. By phenology, I mean the shape of the bloom and how it's structured between the years, and if this could be explained by any environmental variables. And I'll be focusing on the years that were monitored, which was in 2022 to 24 in Nice and Antibes. How I'm going to do that? First off, I'm going to characterize any interannual variation of the blooms that I monitored. And I'm also going to characterize any environmental variables and see if there's also any interannual variation between them. After that, I will combine the two and try to see if I could use these environmental variables to explain any interannual variation of the blooms. The sites I chose, or was chosen for me, was one in Nice. This is next to the port. It's a very busy area. It was sampled in 22, 23, and 24. And then one in Antibes, which is in Salis. It's on the east side of Antibes. And was sampled in 23, but only a few data points in 24. We chose this because it has a very high amount of beach visitors. Like if you go in Salis in June, it's insane how many people there are. So there's a very high risk of contamination by these toxins. For my protocol, I did weekly sampling, or tried to at least. And every time I sampled, I collected 3 seawater samples and 3 algae samples separately. After that, in the lab, I weighed the algae fresh weight and added in my samples Lugol for fixation so I could analyze it later. For the counting, I used the Sediment counting chamber, and I put my biofilm abundances as Ostreopsis cells per gram of fresh weight of algae and my seawater concentrations in Ostreopsis cells per liter. For my analysis, I'm doing 4 big analyses. So first off, I want to test, because I kind of have 2 types of samples. I have my seawater samples and I have my algae samples. I wanted to see if the variation is different, because considering the stock comes from the biofilm, you could consider something different, but because the seawater also has more disturbances, could be different. So I do a linear regression model, and then I test the variation and correlation between the two. Then afterwards, I use the definition that was used in a past paper of a bloom, which said that a bloom is when there's a higher concentration of 200,000 cells of gram of flesh weight algae. And with that, I characterize the bloom peak, start, and end. After that, I'll be doing a time series analysis to really look at the shape of the bloom. But I had a problem, which is my sampling years were different in length. And also, they were not always sampled on the same days, which meant for a time series analysis that I had a very low temporal alignment. So something like Euclidean matching, which is often done in time series analysis, could not really work. As you can see the shape is the same thing, but because it's a bit longer, a bit more stretched out, it doesn't really match it very well. So I used a more novel technique called dynamic time warping. And what this does is it warps one time series to a reference time series, and then with a cost minimizing function, it tries to look and see and determine a distance matrix depending on how much effort it needs to do to warp it. If you want more explanation on that, I can give it later in the questions, because it's quite confusing. Then for the environmental variables, I collected atmospheric temperature, wind speed, and barometric pressure at from a weather station in Nice that was publicly available. And then sea surface temperature I got from satellite data from Copernicus, and nitrogen concentration and salinity were from models. And they all had around the Copernicus had around the resolution of 8 by 8 kilometers. Then I put these in a multiple linear regression model with my biofilm abundances averaged out in log scale to parameterize it. And afterwards, I also did a Tukey post hoc test to test the means of all my environmental variables between the years. So now to go to the results. This was my first analysis, so my correlation between the biofilm and the seawater. So here you see the seawater samples in log scale near the Ostreopsis and biofilm abundances. It was very crazy. The variation looks very off, which is true. But I was able to find a significant relationship with a p-value of 0.3, but as you can see, only 6% could be explained of the variation, giving it a significantly but very weak correlation. This is further proven that the variation was very high by the F-test. It had an F-score of 5.52, which means that the seawater variance was 5.52 higher than the variance in the biofilm. Because of these things, because it was a significantly very weak correlation and the variance was so much higher, I decided for my further analysis to also just completely focus on biofilm because they seemed more accurate of the population of Ostreopsis. Now to go to my blooms. So here I'm having Antibes and Nice, and in this month they were sampled with the different years in different colors. That you can see, I took 23, the Antibes was kind of useless. You're getting a kind of a bloom around the same time at the end of June, and start ending at the end of July for every year, with 23 being the highest peak, and 22, if you look at the bloom definition, had the highest bloom period. And we even started sampling during a bloom, so we probably could even be longer if we sampled before. And 24 had 20 strong typo, sorry. 24 in Antibes didn't have any bloom and had only a week of bloom in Nice. To go to our dynamic time warping results, I'm going to show some alignment graphs, which I don't really have a lot of time to explain, but essentially they're showing what the warping does. It has, for example, for 22, it has here in black the 22 and 23, and the dotted line, if it's very visible, it shows the warping that it's done. If it's vertical, it's because there's a difference in the magnitude of the sampling. If it's horizontal, that means that there's something different in the temporal alignment. So for 22 and 23 had a distance of 2,300,000. 22 and 24 had a distance of 2,900,000, and 23 and 24 of 2,300,000. Now these values look massive because the closer to 1, the more similar they would be. But because I'm dealing in such high magnitudes, they're of Ostreopsis cell, this is actually quite similar. And because they're quite similar, that means that probably there's not any year was an anomaly essentially, because if there was an anomaly, you would expect that one year was very different to all the others, but they're all kind of similar in difference. So there seems to be quite similar alignment between all the years. I'm still under the camera, sorry. For my multiple linear regression model, big word, the R squared was 47%, so 47% of the variance I could explain using my linear model. Sea surface temperature and atmospheric temperature were both correlated, but SST was positive, atmospheric negative. But there was no differences in both variables between the mean values of the years. So there was no variation in the mean between the years. But you could see with SST, like it kind of started to peak a bit later, which could then also explain why in 24 the bloom started much later. Then for my barometric pressure, well, I mean, the R squared is still the same throughout the whole model. There was no significance. And for wind speed, there was a negative correlation with barometric pressure having 2024 being the lowest of all the years, and wind speed having 2024 being lower than 23. and 22 kind of in the middle, non-significant to either of the two. Then to go further to our nitrate and salinity, they were also both negatively correlated. And 24 had lower nitrate on average than the other years, and 23 had lower salinity than other years. Then to go further to our discussion, site variation, there was not enough data collected in ONTIP to really calculate any differences. There was only one full bloom and didn't even really count a bloom according to my definition, so I couldn't do that. In terms of bloom characterization, the bloom timing was quite similar between all the 3 years. They often varied only like 1 or 2 weeks, with only 24 in ONTIP not having a full bloom, but this could also be due to sampling limitations because the variation between my samples, even in the biofilm, was very high. Then in terms of dynamic time warping, there was the main outtake from this is that they were very similar in the results, meaning that no years appears to be a really outlier, and there does seem to be, I say theme because I can't really be sure with like only 3 years, but there seems to be a similar phenology throughout the years in these blooms of 22 to 24. So main takeaway from this is that NICE could expect a year, in every year almost a bloom, and this should be considered as a possible health risk, and there should also probably be a monetary evaluation of this risk because this currently has not been done. We don't really know how much monetary effects this has on both healthcare and economic stuff. For our environmental drivers, this is where it gets kind of crazy because they don't really make a lot of sense a lot of the time. So with SST, this was actually one that was quite nice. It had a positive relationship, which aligned very well with the literature. It was the same mean throughout all the years, which kind of makes sense because in literature, it says that it's more of an onset of the bloom. It kind of kickstarts the bloom, kind of gives it a kick, instead of really showing any impact on the strength. This is maybe why, even though the mean was the same, the timing of the bloom was quite similar, with 24 being a bit later because the SST of the bloom was, SST of that year was also a bit later. But this also has implications on global change, because with rising SST temperatures, this could mean that the bloom starts much earlier. Now, what the real implications of this are, I don't know. Maybe in a way, if it starts in spring and already ends in the summer, this could actually be good for health safety. But we'll see. Of course, this is a very strong claim to make, so I don't stand by this. For atmospheric temperature, there was a negative relationship, which seems incoherent because they're often associated with SST. Perhaps because the temperature and all these variables were taken on the day of the same sampling. You would imagine that an environmental variable on growth rate would have more effect of the days before than of the day the same. If that makes sense. And then with barometric pressure, there was no significant relationship, which is strange because in past literature, it was said that barometric pressure is actually an indication of calm seawater, calm sea. And this is also a factor for algal growth. So you normally, with low barometric pressure, there would be actually more algal growth, which is a limitation of the model. Now with wind speed, sadly, I could not take into account wind direction, which is quite unfortunate because these areas are kind of like sometimes a bit protected from some wind sides, but it was still negative, which aligns with the literature. This is probably because wind can dislodge these cells from the biofilm and also disperse them from the seawater. Perhaps this has any health implications because it also means that they might be able to spread farther the toxins if there is actually a bloom. But this, I could not find anything on or much. For salinity and nitrate, it's probably because they were negatively correlated. It's probably because also the model failed because it's not nuanced enough to use Copernicus data for these coastal microclimates. As I said, I had a resolution of 8 kilometers, while often these coastlines and beaches have a very specific microclimate. Regardless, salinity was found in the past in lab conditions that the highest growth rate was at 37 ppt, parts per thousand. But all my salinity that I measured was higher than that. It was always higher than 38. So perhaps it's not a very linear relationship. And it could be explained that it was actually diminishing the higher actually it goes after this threshold of 37. And now with nitrate, it was very important for algal growth. Maybe it was negative because nitrate is not the only nitrogen, of course, you can find in sea, and it's maybe not as important for Ostreopsis. But if we, for a second, disregard the linear model and we just look at the Tukey test, 22 and 23 had the highest concentrations, mean concentration of nitrate. And these years also have the longest bloom duration, but also the longest peak. So it's not clear, but it could maybe be a slight evidence toward that it actually has a positive relationship. Regarding my limitations, first of all, Ostreopsis and in general harmful algal blooms are very complex. They are driven by a lot of drivers, are often very nonlinear, and I used mostly nonlinear methods, which could have made my results not as accurate. And also they are very different in areas and ecotypes. There's different genetic adaptations that these populations have. Some seem to be good against salinity, some a bit less. So this was also made it hard to compare to literature. There was also, for my sampling method, a very high variation in both the biofilm and the seawater samples. There has been already proposed solutions for this. For example, the Beatty device, which is the benthic, I don't remember, to be honest. It's just a device that makes the sampling less varied, and also something like artificial substrates are being used. And in terms of environmental variables, I was not able to get these coastal microclimates, as I said before. It's actually a low resolution because it's 8 kilometers by 8 kilometers. And I also used, as I said, data on the same day, while all of the papers, they used actually like the SST from 7 days before, because this probably has a higher impact on algal growth than of the day of the same. Well, it has more time to have an impact on the growth. To conclude, the main outtakes seem to be that blooms seem to occur yearly, and this poses a very significant health risk. And monitoring should continue in the area, and outreach should continue to happen of this Ostreopsis blooms. SSC seems to be a very good, important main driver for bloom onset, which is a bit should be monitored, especially with climate change. Also, this SST involves a lot of other environmental variables, for example, salinity. If it's hotter, there's less precipitation and there's more evaporation, so the salinity could increase. So if it's salinity is also negative, that could mean that's like almost a double effect. And also more precise sampling of both Ostreopsis and environmental variables really need to be needed to understand the effect of nitrate and salinity on blooms in the sites. And also something that I didn't write down, forgot to write down, is that I only had 3 years of sampling, which is probably not enough to give a good idea on how bloom phenology works in the area. But still, it's a start, and it was, I think, quite interesting. Then I would like to acknowledge all of the FORESCUE team, Louisa, Aziz, who's also here, Anne-Marine, Sebastian, Florian, Alexandra, and also Victoria. They were really helpful during sampling, but also advice of during the report writing, and also, of course, the Marines coordination team for the bi-weekly checkups. Thank you.
Thank you. So for the reason you just explained, Aziz, who is here, who was also part of your team, is not going to assess your work, but is more than kindly invited to ask you questions anyway. We can start with you, Aldin, going then to Timothy online. But before that, Mila, tomorrow I promise I'll be ready. Mila, your turn. Question.
Does the Ostreopsis cause a long-term breathing problem?
Thing is, we don't know. It seems that the first, once you leave the beach and you have symptoms, after 3 to 4 hours, they seem to go away, like the fever goes away and everything, probably because they get expelled from your lungs. We also don't really know why it makes you, how it's making you sick. And we also haven't really studied yet how it affects you if you get exposed to it maybe like 4 summers in a row, and how that could determine you. So we don't really know as much yet.
Okay.
So thank you for this nice talk. So maybe I missed, how did you perform the sampling?
Yeah, that was maybe not super clear because of time restraints. But so we took in total 6 samples. 3 were algae. So essentially, if I can just show it, we went to a place that had algae, and we took algae, put it in the thing, and then filled with the surrounding seawater.
Yes.
And then the other, we took just surrounding seawater of the close by area. And then in the lab, we measured the weight of the algae, and we made sure to make sure the biofilm also gets stripped off the sample.
But regarding the important topics and the health issue that can. So, are you sure you are collecting at the correct depth?
The study showed that the most effective way to be it was at 0.5 meters. So it was kind of a cheap science, but we made sure to be always like this deep when we were collecting the data.
Okay, so meaning that you want to target site and depth where people can be exposed.
Yeah.
Okay, you want to focus on because you.
And most of the concept is also always at low depth anyway. They're not very. Yeah.
Okay, so it's known that in terms of the biology of the system itself.
Yeah.
So it used to say in really low.
Yeah. It's a low.
Okay, thank you. And so when you are talking about this risk for people, but it's to join a question. So what is it known about the toxicity of this algae and at which concentration it's toxic, or then is there any concentration that?
So are you talking about human or also for other invertebrate species? For invertebrate species, they did try to show a concentration when all of the mortality happened. They did it, like, for example, with some phytal Naupliae larvae. For humans, I also don't know the exact concentration off the top of my head. For humans, it's quite unclear still actually what's making you sick. So they produce something called ovatoxins, which is a derivative of palytoxins. And palytoxins are super dangerous, and in the tropics, they can actually kill people.
Okay.
This is not exactly the same, and this has never happened with Ostreopsis ovata, like people being extremely sick. So we don't actually know yet if it's the ovatoxins that's making you sick, because some have also hypothesized that perhaps it's the theca, like the plates of the dinoflagellates that just get stuck in your lungs, and that's called inflation. That's where the fever comes in. So.
Okay, but the thing is that there is study that show that this algae is also toxic for invertebrates. So meaning that there is not only a human issue, there is clearly an ecosystem issue. Okay, and they know any animal that will not react to this specific species?
Mostly, I think the most affected species are always the less motile. So like the sea urchins and the less affected crabs. So fish, all the time, they don't really. And I think it's also been found in the stomach of salimas, but I don't think they are really affected or anything.
Okay, thank you. And maybe one last question. Is it known for how long a bloom like that can stay? Meaning that you are currently doing sampling. I don't know, maybe I missed the frequency rate.
Every week.
Every 3 weeks?
Every week.
Every week, okay. And do you know how long this high concentration can be maintained now?
Usually, like what I found in the phenology, like they peak, and then once they peak, it starts to go away. So usually the blooms are like 3 to 4 weeks.
Okay.
What happens though is these blooms can come back. I think now, but the fact that it's not really been well studied yet, that there are, and actually now we're also doing that here, is sometimes there's also a bloom in November. If the temperatures are high enough, it can come back in November. So it can come back, but once it starts blooming, then it decreases a lot again. And
okay, thank you.
Okay, let's go to you, Timothée.
Thank you, Ran, for the interesting presentation. So you mentioned that we still don't know a lot about this algae, the toxin, and its effect on public health. However, well, there is a link that is established to some extent. Some effects are known, and then you proceeded to mention that we don't know what the economic effect is. But in terms of prevention and monitoring, what happens? What is the law for such events? Does the beach have to be closed? Do people have to exit the water? And how does it work?
So there's a surveillance network in France, and there's also a big treatment called Ramoge. It's between France, Italy, and Monaco, and also a bit of Spain. And essentially what they do is, in the beginning of the season, the touristic season, they will try to notify any local health workers and also like people like the people who are working on the beaches and stuff about any of the symptoms. And then once they report any symptoms, they say like, oh yeah, people are getting sick on the beach. They will do a monitoring. And if it's after a certain concentration, it will be on alert, on a warning, and then at an alert. And then if it exceeds even more, the beach can actually close.
So the beaches usually don't close, actually.
Depending on the strength of the bloom, really depends on the strength. Because there's I think at 20,000, they are in Ostreopsis per liter. They are in the heavy warning system. So then they will try to do as much prevention as possible. But actually, the beaches closing that much is only in the big cases. Like, for example, this year in Basque, I read that 800 people got sick in one summer. And there they actually had to close beaches because it was getting too much.
Okay, so I guess that in terms of the economic consequences, it's pretty rare that they have to close the beaches.
But there was also, which I thought was a very interesting paper that tried to evaluate any monetary effects, not directly, but tried to do it through a social survey in Monaco. And they did say that people who were aware of Ostreopsis were less inclined to go to certain beaches when there was Ostreopsis. So they hypothesized that Ostreopsis might be more like in the zeitgeist, that people in general will be less likely to go to the beach because they're scared to get sick, which would then also decrease income. So it would kind of be an indirect snowball effect of people just being afraid to get sick because you don't want to go somewhere where you get sick, essentially.
Interesting. So some beaches will be getting bad reputation, I suppose.
Yeah,
one last question. So I guess sea surface temperature will be one of the main drivers in the future. And you said something about how the earlier the bloom, if the bloom gets early in the season, there will be a disconnect between peak tourist season and bloom period. Therefore, we will be less at risk to see. It's a hypothesis. This peaking when tourists are on the beaches or in the water. I'm wondering. I'm not really familiar with this, the phytoplankton or plankton in general, but the winds in the future, how, why does the plankton peak at one point and then decrease? I suppose it's the availability of the fertilizing agents in the water. How are wind patterns going to possibly change this in the future with climate change as well?
So you're asking how changing wind might affect the Ostreopsis bloom in the future, if I'm right. Okay. I know I'm completely spitballing, but I know that winds can often bring a lot of nutrients from, for example, North Africa. I don't know how it's going to change, but I can imagine that these high nutrient, like sand particle loaded winds might actually improve this because it's important for algae growth. And because these algae, they need the Redfield ratio, which is a certain amount of ratio of nitrogen, phosphorus that they want, at least, to grow perfectly. So this might change, but other than that, I don't really know how wind patterns will change in the future. So it's a tough question that I don't really have a large answer for. Sorry.
Yeah, I'm sorry about that question. Yeah, I was thinking more of the thermocline in the thermocline locally in the water, how wind patterns might change and mix the water column in the near future. But yeah, I know
it's a very it's in very low depths so I don't think the thermocline will have that much of an influence then, because thermocline is more in deeper waters, if I'm correct. While this is really most of the Ostreopsis is until you're this deep, you will find, but much more, you won't really find that much more, I think.
Thank you.
Thank you, Timothée. So let's go back to you, Aziz.
Your results. I go to your results.
Which one?
The seawater and temperature correlation.
This one.
Yeah. I think you know the question. Why is the seawater temperature positively correlated, but the air temperature is negatively?
I try to pick my brain over that. I really don't know. I think my hypothesis is like maybe because like the SST is like a kind of a nice increase, and already like because the bloom ends here around most of the times, then it's already decreasing. So probably in the linear model that helps with it. Then here it's often already decreasing when the bloom is still going, because the big bloom is really in this area, which is very variable. So that's maybe my hypothesis on why it's not working. But I tried to look, but it seemed very weird, because they look very similar in their.
It's interesting.
Yeah.
Okay. And just one more question. How do you think your data, and obviously if you collected more data without the cheap science part, data, how do you think this could help predictive models to kind of predict these around the Mediterranean?
There's already actually something very similar that people did to help do a predictive model. They had an R-square of 39, but still, this is a tool that's now being used for like fisheries and aquaculture and also beaches to kind of predict, because Ostreopsis collecting, I mean, you saw me do the data, it's very time intensive. You need to go to the beach. You also like the counting takes a very long time, especially if you're dealing with high concentrations. So if you could have these very simple parameters that even like to Copernicus, they're often available like the next day sometimes. That would be, if you could generate a model where you could predict, oh, now there's a high chance of there being a bloom in this area, that could be very interesting to know where you want to start counting Ostreopsis, because right now they're relying on, oh yeah, if you notice any symptoms, then we can start counting. But it would be very much better if you could know, like before the symptoms start, that we could start counting. Thank you.
Thank you. Just maybe I have two questions, just because as it cannot evaluate you. I would like to know what's your definition of a bloom first. And as a second question, you're talking about phenology and you're saying that it's quite the same phenology from one year to another. So my question is, what could have been a different phenology according to the literature you read and according to the specific conditions here in the sites? What could have been a different phenology?
I would say different phenology would be just no bloom, just not having not a bloom or a much later bloom, much earlier bloom. And also, well, to go to your first question, the definition of a bloom was if the biofilm samples had a higher count than 200,000 Ostreopsis cells.
Yeah, but why?
It's very, very subjective. I had a very long discussion with it, and it's very hard because a harmful algal bloom is also from a health context. I think they determined that because 200,000 is used in warning systems, and it's also a time where it starts getting dangerous for people.
Okay.
Because the bloom is mostly used, the harmful algal bloom is mostly used in a health context, actually.
Yeah, but the time of a bloom is related not to health issues. Most of the time, it's just like phytoplankton blooming.
Yeah, but it's very hard to then know, like, okay, like, technically here it's already blooming. So maybe you can look at the growth rate, but it's and also then you have the variation that's very high. So sometimes it's dropping. So when it's not blooming anymore, it's quite hard to know with this type of data, monitoring data. So that's why they chose to have a very simple, if it's above 200,000, that's the definition.
Okay, good for me. So we can open the questions to the audience, and in the audience, hello, Emma. You had a question? Emma Benlamin from MedFund.
Yes, hi everyone. Thank you for your nice presentation and clear explanations. I have rather a curiosity question. If there are studies that focus on the transmission of Ostreopsis in the trophic chain between prey to predators or predators to others.
I think there was one study that did like on toxic, I think there was one big study still only on toxic accumulation of this species, other Ostreopsis species, but they're much more toxic. So there's a higher relevance there. But they did, I think, mostly sea urchins, but also they looked at some types of fish and species, but they did find toxins. But it's very hard to, because we don't know the thresholds yet for humans. So therefore, like to measure like an LD50, it's quite
Okay.
hard.
It's something that can be transmitted from a
Yeah.
prey to a predator.
I know actually, if you know Maria Yasmin,
Yes.
see, her PhD student is studying that. So they're looking at from the tiniest shrimp to also the big fish and how much like Ostreopsis cancer or isotope analysis they have.
Mhm.
So they're actually working on that right now to see how the trophic chain is going through that.
Great. Thanks for answering.
That's time for a couple of questions also from the audience. Yes.
First of all, you warp time, so that's very cool. So well done. But my question was just, and sorry if I missed it, but when outside of bloom situations, like in the winter when it's cold or whatever, is there still like a background little quantity of Ostreopsis in the water, or is it like virtually none and then a lot?
There's always some, so they have something called light cells and dark cells. So they have a certain amount of cysts. They have a cyst, so they can survive less favorable conditions. So they always have kind of a cyst population. And then when the conditions get right, they start, their cyst opens and they start proliferating again in their normal form. That's
I want to do a stats question, if that's okay. When you did the multilinear regression, right, you showed that some of them are significant and some of them aren't. But did you look at the magnitude of it with the beta, with the coefficient, or
the estimate, you mean?
yeah, with the estimate?
Yeah, I looked at the estimate. I just didn't include it here, but I have it in an annex.